As I was saying, welcome to today's webinar. My name is John Cadiz. I'm one of the PIs that help to coordinate HIRN. And if you haven't been to one of these webinars before, what we do is we try to highlight some of the technologies and some of the science and the data that has been coming out of this network or through collaborations with this network. So we have a great team today that has assembled a wonderful talk. For those of you that aren't aware of HIRN, HIRN is a network that was established back in 2014, and the structure of it was that there were different themes that were targeted. Each of these different circles represents a different foci. One of them, the consortium on beta cell death and survival, another consortium on targeting. And so each one focused on a different aspect of type 1 diabetes, but over the years it has changed. And what it is currently now is it's a group of 4, soon to be 5 different consortia or groups working together. And I'll give you a little bit more information on that shortly. But in total, over the last 10 years or so, we've had anywhere in the neighborhood of 800 different researchers from a lot of different institutions working on type 1 diabetes, working on different diseases, working in different disciplines, collaborating together and working at different problems. If you want to be connected to a lot of this, we have a website which I'll show you, but there is a meeting coming up. It's a 10-year anniversary meeting organized by HIRN. It's happening actually on the NIH campus, so you can register for that meeting. It is going to be in January. And there is a venue that you can check out there, but there's lots of other ways if you want to stay connected as well. We do have a website that we try to keep up to date. It talks about all the different consortia and groups, different funding opportunities and different awards that are coming out. If you're looking for jobs, if you're a postdoc maybe, and you're looking for a lab to join, you can look at job opportunities there. Different databases that you might be interested in, different data sets that you'd want to analyze or tissue that you want to get. You can do that on the website as well, too. Of course, we have a number of different media, social media outlets. And then what we do is we also maintain a catalog of different antibodies and cell lines and tissues and protocols, and you can find all that on the website as well. And we also keep track of the different webinars. So today will certainly be interesting. You won't have to worry about writing everything down because it will be posted shortly after today, and you'll have that webinar available to look at your convenience. So with that, I'll just remind you to keep yourself muted. We've got a great number of topics that we're going to talk about today. And so what I'm going to do is I'm going to hand off the webinar to our first speaker, Dr. Raghu Mirmira. Dr. Mirmira is a professor of medicine at the University of Chicago. He's also a clinical scientist that I have to remind myself of. He's a board-certified clinician in endocrinology, diabetes and metabolism, in internal medicine. He directs the Diabetes Research and Training Center at the University of Chicago. And if he's not seeing patients, he's maintaining quite a prolific lab of over 20 individuals. He's an islet biologist. He's interested in the pathogenesis of type 1 diabetes. And so he's into a lot of different things. And with that, Raghu, if you would like to take it from here. Raghu, you're muted, so you might want to unmute yourself. Or perhaps I'll unmute you. There you go, Raghu. I can hear you now.
Okay, all right. For some reason, my controls were missing. But
No
anyway,
problem.
I'm back. All right, wonderful. Thank you so much, John. That was a really kind introduction. And while you introduced me, I'm actually going to introduce some smarter people than myself who really can teach you a lot about extracellular vesicles. I run one of the CBDS awards, and two of the people that have been involved in my award over the years are Ernesto Nakayasu and Emily Sims. And we've been, among the things that we've been studying is our interest in extracellular vesicles and their communication with other cell types in the context of type 1 diabetes. I wanted to just highlight this particular HIRN review article that we published now almost 2 years ago, where we talked about really sort of the role of lipids and how extracellular vesicles are formed, how they incorporate specific lipids into their structure, and then how those extracellular vesicles then communicate with other cell types. So just a couple of basic things about extracellular vesicles. There are different types of extracellular vesicles that can emanate from any cell, so not just a beta cell, but whatever cell type you might be interested in. And the one classic extracellular vesicle you might hear a lot about are exosomes. They form from sort of the endosome pathway. And these endosomes, have multiple intracellular fates, one of which is to form these multivesicular bodies where they incorporate a variety of different cargo. And you can see what some of that cargo could be. It could be typically microRNAs, RNAs, proteins, and a variety of metabolites. And one thing you'll learn today is that it's not just a random incorporation of this cargo. Some of this stuff seems to be actively transported, for example, into these exosomes. And then these exosomes can then subsequently be released. And then I'll tell you a little about what they do. But there are other types of extracellular vesicles that can emanate. And just a couple of other types are microvesicles. These form from budding as opposed to the endosomal pathway. And they can also incorporate a variety of different intracellular cargo, including metabolites and proteins and nucleic acids. They tend to be much larger, and we define them largely by size. You can see here they can extend up to 500 nanometers in size. And then there are even larger extracellular vesicles called apoptotic bodies. And these can incorporate, including everything I just mentioned, also DNA, and may reflect more sort of the apoptotic process in these cells. So the interesting aspect of extracellular vesicles is their communication with other cell types. So they can communicate in the local environment, they can communicate more systemically as well, and they can communicate with, for example, the immune system, which is something in particular we're interested in, but they can also communicate with neighboring cell types like other beta cells and alpha cells. And so, in our review article, we talk about sort of how some of these, in particular, exosomes, which is one that we've been particularly interested in, are formed in terms of their lipids, and then in terms of the kind of cargo that can get incorporated into these otherwise large multivesicular bodies, which then get released once they fuse to the membrane as exosomes. And these exosomes can then, in a neighboring cell, be taken up, and then that cargo can be incorporated into that cell. So this is a different means of communication, different than hormone signaling and so forth, which means that you could literally get nucleic acids from one cell type into another, and that can affect the ultimate production of proteins in that other cell type. So really, without much further ado, I want to introduce our two speakers, and they'll tell you a little bit about the formation of these, the cargo, and how these can be used arguably as biomarkers as well. And so our first speaker, I'm going to introduce, I'm going to stop sharing, and then I'm going to let Ernesto take over, is Ernesto Nakayasu. He's at the Pacific Northwest National Lab. And Ernesto and I have been collaborating for the better part of 10 years now. He's currently a scientist level 5 and leader of the biochemical mechanisms team, but he's soon to be getting a promotion to become the leader of the multi-scale systems science group at PNNL. And he's going to talk to us about extracellular vesicles as biomarkers for type 1 diabetes. Ernesto, take it away.
Yeah, thank you for the kind introduction, Raghu. Are you seeing the right window or?
No, the wrong one, Ernesto. Go ahead and swap it. Yeah. Okay, perfect.
Okay. All right. So today I'll talk a little bit about how we are targeting using vesicles as biomarkers. So if you think about the extracellular vesicles, they send signatures, or communication to other cells. In our case, the islets, they send communication to immune cells and receive it as well. So you have some signatures of disease, and we want to get the signatures to see if they can be good biomarkers. But there are some challenges of doing biomarker study in vesicles. First is contaminants. As you can see, we usually want the main samples that we use for biomarker studies, plasma, and in plasma is very rich or other particles that are similar in size and other physical chemical properties to vesicles, such as lipoproteins and protein aggregates, and even things like viruses. The other problem is the volume, because usually most of the approach to purify extracellular vesicles, they require a lot of volume, like 1 to 2 mLs of blood. Even if you think like, a kid, like how much blood you can get from them. So 1 and 2, if you target to get samples from consortia, 1 to 2 mLs is like really impossible, because that is the whole thing that they have collect for to doing dozens of different analyses. So our approach is a little bit different. So instead of like really be able to purify these to a great extent, we're thinking of our approach is to understand what is the composition. Can we deal with some of the impurities and still get the data that we need? And the other part is we need to maximize the recovery, so we don't need to have to use as much sample. So to understand the composition, we did a new approach here. We we did something called meta-analysis. So we got the data. These are not data that we collect, but data that other people collect and left available through different portals. So we went to the mass spec portals, the PRIDE and the Massive, and we got data from 7 different studies. And the nice thing is that they come from they use all different methods for purifying the vesicles. And the idea that we have, well, if they have their different methods, we have different proportions between vesicles and contaminants, and maybe we can use those to kind of cluster them out and separate between them. So we analyzed the whole data set together, and we actually got quite a massive number of proteins. We usually get about, when we look at the whole plasma proteomics, we usually get less than 2,000 proteins, and we got about 4,600 proteins in this meta-analysis. So then we cluster this. So when you cluster this, so the idea is that we can cluster the contaminants separate from the vesicle proteins. Then we have 2 clusters. So this cluster 10 and 11, we see that they have a lot of EV markers. In this cluster, they have about 1,700 proteins. And the most important it was that we were able to validate this. So we did a systematic review of the literature, and we looked for things that were previous validated by ImmuneGold, and we used those proteins to calculate the true positive rate. So we had this true positive rate of 71%. So it means that these two clusters were enriching this, really enriching this AV protein. So when we look at them to know about non-contaminants, we see that the seralbumin is a common contaminant. They cluster completely different in different clusters. We also see that lipoproteins is a non-contaminant as well. You can see that they cluster in different clusters as well. So then we start to validate at least one of the new identified proteins. So Raghu lab did this experiment for us. So it's called NanoView. In the technology is this. So you have these microchips that they are coated with antibodies against the EV markers, CD8, 63, and 81. So you use those to capture the EVs, and then you use secondary antibodies to measure your specific protein. And the protein that we were interested was protein basic protein, because this in other studies we found that was a biomarker for type 1 diabetes. And then you can see this, that not all the vesicles, but there is certain percentage of vesicles that you can see here. They're positive for PPBP, or protein basic protein. And here's just the quantification to show, so it's a small percentage, but the PPBP is in extracellular vesicles. So now we can understand better, because if you look at the literature is a little bit messy. There are people claiming all sorts of different things in the vesicles, but now with all this refined data set, now we can see what is really in vesicles or not. So if you look at here in cluster 10, 11, we are very rich in cell-to-cell communication proteins, also intracellular signaling, lipid-mediated signaling, and some of the metabolic proteins. These are very classical known proteins for to be in vesicles. But there's some of the things that were not very consistent in the literature. Things like ribosome or spliceosome. When we saw that they come in a different cluster, we think that these are more contaminant, because if the cell bursts, and release the ribosomes, the ribosome particles are about the same size of some of the small extracellular vesicles, and they also have similar density as well. So then, if you do ultracentrifugation, something like that, then we would also capture those. We can see that they have a lot of different cell markers. So then you can try to think where they're coming from. There are a lot of things like erythrocytes, a lot of the immune cells. In plasma, of course, platelets is a big thing, and then some of these co-purify too. But the other thing that we look at were the CD markers. And then if you can see here, CD markers very enriched in the cluster that are the extracellular vesicle clusters. And you can see basically 90% of all the CDs that we identify either in cluster 10 or 11. And then we think that, because CDs are not going to be just floating around, they need to be bound to some kind of membrane. So that's the way that they come out with extracellular vesicles. Okay, so then now we try to switch to our next stage to look at extracellular, to look at a good method to purify vesicles. And I put here the different methods again. And as we can see here, the panel A here are how much of the proteomic fraction was each of these non-contaminants, so seralbumin, apolipoprotein. And we can see that some of the methods are, 2/3 of the protein we identify is based, the protein mass is contaminant. So this polymer precipitation, for example, is mainly enriching for lipoproteins, not for vesicles, because actually they bind to lipids, which the lipoproteins are more enriched with lipids than vesicles. But here in the bottom here is the recovery of the proteins in cluster 10 level. So those are the EV proteins, and we can see that C here is centrifugation, but this is a centrifugation at lower speed at 10,000 Gs. So we think that those are mainly like the larger vesicles, and size exclusion chromatography also has a very good performance. So we decided to go to size exclusion chromatography because we thought that would be less biased, and because we can also automate the process, then I think it will be more reproducible. So we made this size exclusion chromatography. We instead of like the gravity one that you can buy and the kit from QEV, we did it on a FPLC. So then we can do that automated. So they have an auto sampler, an injector auto sampler, so you can do your samples there and to run them. The nice thing is we use a small column that is only 15 centimeters long and half centimeter in diameter. So we don't need as much sample. We can only use 50 microliters of plasma in our analysis. And because it's also a short column, we can get the samples purify only 15 minutes. And as you can see here, it's very reproducible. Here is 3 replicates, and they have R square that is over 0.95. Here's how the chromatogram looks like. So this looks just to be just one, but actually there are 3 different chromatograms, one top each other here. And you can see how reproducible the chromatography is. In this panel here, we show that all the still well characterized EV marks, they come as this fraction 3 here. So we see that they can enrich for the vesicles in a very fast way, and also that it's very sensitive. We test the system first because we didn't want to get very say special samples, and we had this from a previous study that we studied firefighters, that they go for a training, and we had this sample sitting around. So we were trying to understand like how their body change, how their body respond to the intensity of their duty when fighting wildfires, and as you can see here, we had 9 samples pre and post exercise. It's kind of a little bit hard to see, but 6 of 9 we actually have higher EV peak after exercise, and then you look at different pathways, then we can see there are pathways that are well known for that are activated during exercise. So it means that the system is working well. So we move now to look at extracellular vesicles from donors that are antibody positive, auto-antibody positive. So those are samples that Emily sent us, and we see here that we still have very reproducible chromatography. And when we look at this, so here we are comparing the protein with the whole plasma proteome that we did for TADI. So TADI stands for the environmental determinants of diabetes in the young. So we conduct a very large biomarker study there. And here you can see that the overlap is not very big. So it means that running the EVs now, you can get a new set of biomarker candidates and see when the ones that are significant, also there is even less overlap. The pathways we can see here in the regulate EV proteins are all kind of related to inflammation. So I think it's probably signals from the insulin process. And let's see what I have. Yeah, so that's where we are now. And so just the conclusions from this talk. We were able to distinguish, like in our meta-analysis, what are EV proteins or what are contaminants. So now we can just use a method, because right now we know what are the EV proteins, so we can just need the method now to quantify. And then, so we are using this size exposure chromatography that we can only use 50 microyears, and we can just go and quantify this protein. We also identified some biomarker candidates that we are now, we are now pursuing to validate these in a new set of samples. So I have a lot of people to acknowledge, but especially I wanted to acknowledge Mileni, who did most of the meta-analysis, in Ivo, who is doing all the size exclusion chromatography. We have our collaborators, Emily, Raghu, Sasanka from, and the big team here from Philippines now, and thank you. So now I'll turn this back to Raghu.
Thank you, Ernesto. I think we're going to do questions at the very end, and I'm now going to introduce Emily Sims. And Emily, you can load up your presentation. Emily is an associate professor of pediatrics at Indiana University and has been a collaborator of mine for over a decade now. And she's going to tell us about a new study that was just recently accepted as well here into Diabetologia, and I'll put a link to the preprint of the paper in the chat. And she's going to tell us about beta cell extracellular vesicle PD-L1 and its role as a regulator of CD8 T cell activity. All right.
Thanks, Raghu. Do you guys see the right screen for me? Is it the?
Yes, we do.
Okay, excellent. Thanks for the introduction. Thanks everyone for your attention. I'm Emily. I'm an associate professor at IU and a pediatric endocrinologist. And then here are my disclosures. And I actually, sorry, Ernesto, your picture is different, but I actually really wanted to show this slide because I want to make sure I disclose that I am really lucky to be working on this as a very collaborative project with a group of really amazing and fun to work with investigators on this grant. So this is work from many great minds that I've had the opportunity to work with. And I also especially wanted to highlight the person on the far left, Chaitra Rao, who's a really talented postdoctoral fellow in my lab who did the work that I'm going to show today. Okay, so Ernesto and Raghu did a really great job of introducing the idea of EVs for me. So I'm not going to stay on this slide for a long time, but just to say that my research program has had a big focus on these molecules. We're especially interested in them for their biomarker potential because, as mentioned, the molecular cargo within these EVs changes based on the health of the parent cell, and they're very stable in extracellular fluid. And as you guys all know, we need better biomarkers of what's happening in the islet microenvironment as type 1 diabetes develops. And then we're also really interested in understanding the physiology and the pathophysiology of what EVs are doing in health and disease in terms of how they're allowing cells in the islet microenvironment to communicate. Okay, so background for today, this project is focused on PD-L1 and EVs. And so some background to PD-L1 or programmed death ligand 1 is that this is part of this class of protein called immune checkpoints. And so for people who aren't familiar with immune checkpoints, these are molecules that are basically on the surface of all cell types. When I first heard about immune checkpoints, it was in the context of tumor cells or cancers. And the idea is that these proteins like PD-L1 or programmed death ligand 1 sit on the surface of a cell, and then they interact with their receptor on immune cells to deactivate the immune cell. So the immune cell doesn't recognize the original cell as foreign and doesn't destroy the foreign cell. And so in the context of tumors, this is a way that tumors are able to kind of evade the immune system and evade immune surveillance. But we've all heard of checkpoint inhibitor therapies, which are a type of medications that have really revolutionized cancer treatment because they allow somebody's own immune system to recognize tumor cells or cancer cells and attack those cells and destroy those cells. But interestingly for me, as an endocrinologist, a well-known side effect of this class of medications is development of these autoimmune endocrinopathies, including a form of diabetes that looks a lot like type 1 diabetes and is an autoimmune diabetes, and suggests that these checkpoint molecules are an important way that the immune system interacts with the beta cell. And specifically PD-L1, inhibitors of PD-L1 are one of the types of drugs that are associated with this autoimmune type 1 diabetes. And so multiple investigators, including Desi Oziric, who's one of our wonderful collaborators, have shown that PD-L1 is present in the beta cell. And so these are some data from his group showing that if you look in human islets, PD-L1 is there, and it's actually upregulated by exposure to interferons, which we know is really important, especially early on in the context of type 1 diabetes development. And if you look in islets from people with type 1 diabetes, there's upregulation of PD-L1 in the residual insulin-containing islets when you compare them to control islets or to islets that no longer make insulin. So again, this is supportive of this idea that an increase in PD-L1 is maybe protective to kind of help the beta cell evade destruction by the immune system. And so we were really interested in if EVs could play a role in this interaction. And so specifically, Chaitra asked if PD-L1 could be present in beta cell EVs, and if this could be a way that the beta cell was interacting with the immune system to kind of prevent its own destruction as a protective mechanism, so that beta cell EV PD-L1 would decrease immune cell activation and ultimately decrease beta cell destruction. And so to do this, she isolated EVs in a couple of different ways, depending on what our readout was going to be, including ultracentrifugation and size exclusion chromatography, which we've heard about already. And as a first step, she really just wanted to see, okay, is PD-L1 in these EVs, and is it upregulated in the same ways that we see it's upregulated within the beta cells? So here she performed Western blot in both ENS1 beta cells and Indo-CBH1 beta cells, and looked at cells and the EVs from those cells at baseline and in response to interferon alpha treatment. And what she found was that, consistent with the knowledge we already had, PD-L1 was upregulated in the beta cells in response to interferon alpha treatment. And we were very excited to see that PD-L1 was also really, really easily detectable and present in the EVs, and that this was upregulated also by interferon alpha treatment, similar to what we saw in the cells. And the other proteins that are on this Western blot, you've heard a little bit about some of them. CD63 and CD9 are both tetraspanin proteins, which are molecules that are associated with EVs or on the surface of EV membranes. And so we usually check to make sure those are enriched for in these EV preparations. And then calreticulin, as you know, is an ER protein. So we check to make sure calreticulin is depleted when we do these EV Western blots. Okay, so these are data from cell lines, but we also looked in human islets where we saw that PD-L1 was significantly upregulated with treatment with interferon alpha. And the graph on the left side of the screen is showing nanoparticle tracking analysis to understand the changes in total EV number with interferon alpha treatment. And so here you can see there's really not a difference compared to control. And so what this told us is what we're measuring when we measure this increase in PD-L1 isn't just this kind of non-specific increase in the number of EVs so that PD-L1 is increased, but that it's a specific shift in the cargo of PD-L1 within the EVs that are being released from these beta cells. Okay, so, we really want to know if PD-L1 is doing anything and engaging with its PD-1 receptor when it's on these EVs. And so an important question is it on the surface of the EVs or is it within the EVs? And so to ask if it's present on the surface, Chaitra performed an EV isolation using these ExoView EV capture chips, which Ernesto talked about a little bit already. But just to review, the idea here is that you do EV capture that's antibody-based with these surface tetraspanins, and then you can detect the presence of an antibody to a protein of interest on the surface of the EVs. And so here we did this for PD-L1, and we found that indeed, when we looked at the kind of most common tetraspanins for EV capture, PD-L1 was upregulated on the surface of these human islet EVs with interferon alpha, and actually we also tested with interferon gamma, and they both significantly upregulated beta cell EV surface PD-L1. So as a side note, we wondered, okay, so PD-L1 is increased. Are other proteins that we know are induced by interferon gamma in the beta cell also affected in these EVs? And so I thought this was kind of interesting. Chaitra also performed detection antibody staining with HLA, with a pan-HLA antibody. And we also found that there was an upregulation with interferon alpha or gamma treatment of HLA on beta cell EVs in concert with PD-L1. And here's the quantification of the upregulation for HLA on the left and PD-L1 on the right. And so again, you can see they're both upregulated, and actually, especially with interferon gamma, the HLA were really, really upregulated compared to the PD-L1. But we wanted to know, is this on the same EVs? Because I kind of think of these as good and bad. So it seems like they're kind of working in opposite directions. And so she also performed a co-localization analysis within these EV samples and found that actually it's really unusual for the HLA to be detected on the same EV as PD-L1. And we wanted to make sure this wasn't, something like steric hindrance. And so as a control, we also performed co-localization analysis for one of those tetraspanins, CD81. And in contrast to HLA, really commonly we found that HLA and CD81 were co-localized, but the percentages of EVs that were positive for PD-L1 and HLA together were really, really low. So this suggested to us that these were kind of separate EV populations that these cargo were going into. All right. So as I mentioned, we were really, really excited about understanding the potential functional relevance of what this PD-L1 on these EVs were doing. So, I already showed you data that it's present on the surface of EVs, but then the next question is, can it actually bind and engage with its receptor PD-1 to have an effect on immune cells? And so to first get at this, we performed a binding assay. This used FRET-based technology, and we did this with Endo-C EVs. And so you can see if you put PD-L1 standard into this assay, you see this logarithmic curve demonstrating binding to PD-1. And we saw the same thing when we added EVs from Endo-CBH1 beta cells. And as a control, we also looked at HEK293 EVs, which have very low endogenous PD-L1 levels. And so you can see with the gray line here that when we added these into this assay, we really didn't see very much binding. We don't see that logarithmic curve. But if Chitra overexpressed PD-L1 to increase PD-L1 expression in those cells and then use those EVs, again, we were able to restore this logarithmic binding through this assay. So this suggests that indeed this PD-L1 on the surface of these beta cell EVs is able to bind to PD-1 in this PD-1 binding assay. All right, so next, again, just really trying to get at this physiologic impact. We were excited to do some experiments to see what we could do to immune cells. And so for this work, we were able to collaborate with John Piccinelli, who's at IU now, to do some immune cell treatment experiments in a postdoc in his lab, Dr. Roy. And here we took splenocytes from NOD mice, and we got their splenocytes out and we stimulated those splenocytes with CD3 and CD28, and then did a bunch of different treatments with different kinds of EVs. And we used EVs from NIT1 beta cells, then performed flow cytometry for CD8 cells, and quantified different measures of T cell activation, as well as T cell effector responses in the supernatant. And so I'm just going to walk you through the controls and then the actual graphs from the experiments, because there are a lot of controls in these experiments. So one of the types of treatments we did was just with wild type EVs from those NIT1 cells. Another experiment we did was using EVs from those NIT1 EV cells, which have pretty low endogenous PD-L1 as well, but we overexpress PD-L1 to make sure there's lots of PD-L1 in the EVs coming from those cells. Another experiment we did was with those PD-L1 overexpressing EVs with a blocking antibody to see if we specifically block PD-L1 on the EVs. Can we block the effect of those PD-L1 overexpressing EVs, as well as an isotype control antibody to control for that blocking antibody? So 4 different controls here. And so here's a graph looking at the effects on proliferation in those CD8 T cells. And so here we looked at cell trace violet to quantify proliferation. And you can see the graph for each of the groups, which I'm going to walk you through in a second. And these are quantified on the right side of the screen. So in the gray line at the bottom, you can see unstimulated CD8 T cells, which don't have very much proliferation. And then the black line up here shows when we stimulated those cells with CD28 and CD3, the proliferation increased as we would have expected. However, and also when we add just wild type EVs from those NIT cells, which have pretty low endogenous PD-L1, we didn't see much of an effect on proliferation. If anything, it was actually a little bit increased by those wild type EVs. However, when we add the PD-L1 overexpressing EVs, we're able to reduce that proliferation. And this didn't change when we had the isotype control antibody in there, but that effect was blocked when we added in anti-PD-L1 antibody to block the effect of the PD-L1 on the EVs. when we looked at a marker of early activation, CD69, we saw really similar results. So gray line at the bottom is unstimulated cells. When we added CD28 and CD3, we saw a big increase. That's the black line. This was not really changed by wild type EVs that much, but we saw a big decrease when we added the PD-L1 overexpressing EVs, which was abrogated when we added in the anti-PD-L1 antibody to block the PD-L1 on those overexpression EVs. And then we also saw similar relationships when we looked at a marker of late proliferation, CD25. Again, unstimulated cells in gray. The CD25 really increases when we stimulate those CD8 T or those splenocytes, but then this is reduced by the EVs with PD-L1 in them, and we're able to block that effect of the PD-L1 EVs if we add antibody in to block PD-L1. So as I mentioned, we also wanted to look at effector responses, and so we measured granzyme and interferon gamma in the supernatant, and we saw really similar patterns here as well, where in gray we have unstimulated cells, in black we have stimulated cells, so the production of these were really increased, but then this was reduced when we added in EVs that had that PD-L1, and that effect was blocked when we pre-treated those EVs with an antibody to block the PD-L1. Okay, so I think the immune cell data are very cool, and these are our lab's kind of first forays into beta cell immune cell interactions. So we were really excited to be able to show that effect. But also, we're always thinking about translational relevance to humans. And so we also wanted to do some testing in human samples. And so these are data looking at EV-PD-L1 from a group of children with new-onset diabetes from the Children's Hospital where I work, compared to a group of non-diabetic controls. So I was super excited to test these samples, and then very, very sad when we saw that there weren't any differences in the plasma EV-PD-L1 in the children with new-onset diabetes compared to the controls. But then we kind of thought about, okay, well, maybe this is related to heterogeneity that we see within phenotypes of type 1 diabetes. And so we looked at some different measures within the group of children with type 1 diabetes. And what we saw that we thought was really interesting was that there was a positive association of how much PD-L1 was in the plasma EVs with the residual C-peptide in these children. So the higher your plasma EV PD-L1 was, the higher your residual C-peptide tended to be. And this relationship was not present in the group of non-diabetic controls. And so we thought, geez, this is really interesting because it kind of parallels the data that Decio's group published before in islets from people with longstanding type 1 diabetes, where there was an association between increased PD-L1 staining and residual beta cell insulin positivity. And so we also looked in samples from people who didn't have diabetes but were islet autoantibody positive. And here we thought it was really exciting that actually the EV-PD-L1 in plasma was higher in the autoantibody positive controls, or excuse me, the autoantibody positive people compared to autoantibody negative controls. And what was really, really interesting about this group is that there was a mix of people who were single and multiple antibody positive. And my kind of hypothesis had been that the people who were multiple antibody positive would have the highest EV-PD-L1, but it was actually the opposite. The squares here show the single antibody positive individuals, and these are small numbers, right? But those are the individuals who overall had the highest EV-PD-L1 compared to the controls and the people with multiple antibody positivity. And again, when we looked at C-peptide within this group, there was this correlation with C-peptide levels and EV-PD-L1, where if you had higher EV-PD-L1 in your plasma, you had a higher C-peptide only within the islet antibody positive group. That was not the case for the non-diabetic controls. There were no relationships between those measures. All right, so to summarize the data I've shown you today, we've shown that beta cell EVs carry PD-L1 cargo on their surface and that it's induced by interferon exposure. I think the HLA data are kind of interesting and suggest that, this isn't cargo that just kind of goes into all of the EVs, but I think it suggests there's some regulation in different beta cell EV subpopulations that contain PD-L1 cargo. Additionally, our functional data suggests that beta cell EV-PD-L1 binds PD-1 and reduces CD8 T cell activation. And that circulating EV-PD-L1 is increased in autoantibody positive individuals, particularly those who are single antibody positive, and is positively correlated with C-peptide in people both with autoantibody positivity and recent onset diabetes, type 1 diabetes. So, there are certainly some limitations to this analysis that I showed you. The plasma samples were cross-sectional. And also, in plasma, EVs are coming from lots of different places. I know a lot of people in this consortium have been working on identifying beta cell specific or beta cell derived EVs in plasma. It's challenging. These were just kind of cross-sectional plasma samples. So, it could be that we're measuring EV PD-L1 from many different cell types. We probably are. But I think that it still could be that, a person is predisposed to having increased PD-L1 in their EVs from lots of cell types. And this is also reflected at the level of the beta cell. And that our data suggests that there is a role for EV PD-L1 in beta cell survival, both from autoimmunity and also in the heterogeneity that we see in C-peptide loss and type 1 diabetes and its progression. So next steps, we're really interested in these functional impacts, particularly in other immune cells. What are the other kinds of cargo in these PD-L1 positive beta cell EVs? And then also, could there be effects also on other beta cells? So we're looking at beta cell transfer. We've been working on developing an iPSC line that has labeled PD-L1, so we can more effectively and elegantly track the transfer. And we got approval to test primary tissues in pod slices, the perifusate, for people with T1D. So I think that'll be really interesting compared to controls. And then finally, I think it'll be really important to test longitudinal samples in auto-antibody positive people, particularly single antibody positive, and understand relationships with diabetes progression in these individuals long term. So I'd like to thank our funding sources and all of our great collaborators and the participants and donors. And that's the end of my presentation.
Thank you very much, Emily. That was great. And Ernesto, if you can join too, that's wonderful. Yeah, thank you all for that wonderful presentation. I'll invite the audience, if you have questions, unmute yourself, raise your hand. I'll be looking at the participant list here and calling out questions in the order that I see it. Jay, do you have a question? Yes, go ahead.
Yeah, this is, I was pretty excited to observe Emily's work. Very fascinating, particularly the context of type 1 diabetes heterogeneity. My question to you, do you think that a different responsiveness towards anti-CD8 therapeutics, particularly the biologics, may have some kind of link with the hypotheses that you are pursuing?
Yeah, that's a great point. I mean, I think that for sure there could be differences in, the amount of these EVs people make, maybe in the ways that the EVs react. Yeah, and it could totally affect treatment response. I think that's a really good point. It could be that, maybe some people, I think it could be in multiple ways too. It could be that maybe some people are already making lots of PD-L1, right? And so they don't need something that's going to kind of shut down T cell killing, like in the acute phase, or it could be that those are people that don't have it, that are like right for treatment, and might be more responsive. Yeah, it's a really, really great point. We should look at that at treatment response, I think.
Thank you, Jay. Wei-Zhang.
Thank you, Emily. I think it's a very interesting data on PD-L1, and it's quite compelling, like a PD-L1 play a role in the islet. But I was really stuck by the plasma data or serum data. So how could the PD-L1 increase just in those auto-antibody positive or correlated with residual C-peptide? Because islet population in the body is such a small population, right? And it's very hard to understand PD-L1 is actually come from beta cell. So could it be just like this global PD-L1 regulation, and it's linked to this whole mechanism, not only beta cell, basically like those individual, right? Maybe there's a protection mechanism globally.
I agree with you. I totally agree. So I think it could be, certain people have this global response where they're making more PD-L1, right? And we know there are systemic inflammatory signatures and stuff early in type 1, right? So it could be that you're seeing a global upregulation response to that, right? But it's just that's also present at the level of the islet, right?
Yeah. Okay.
Yeah.
Thank you.
Yeah, thank you. That's a really great point.
Thank you, Wei-Zhang. Hi, Decio.
Thank you. Well, can you hear me?
Yes, Decio.
Okay, well, this was a very interesting seminar. Congratulations to the speakers. one thing that would be very interesting would be to be able to capture the exosomes that are specifically released by the beta cells. And I wonder if you could check if these vesicles carry DPP6 and FXYD gamma 2A, because these are two biomarkers that we have discovered, and we have nanobodies or antibodies against them. So if these proteins are present, we could fish them, and then you could work specifically with beta cell-derived vesicles.
Yeah, I think that's a great point. We've had a little bit of a challenge with specificity, like of the control antibodies with those pull downs has been a challenge for us. But I think the FXYD2 protein is there. So if you guys have a great antibody, like it would be great to try that. And then I haven't tried the other one, but we could certainly look, Decio. I think that's a great point. Yeah, because it would be it's a goal that we're all really interested in, right? I think. Sorry, go ahead, Ernesto. I cut you off.
We're just going to say that Milene checked for those proteins in the plasma data that we had, but we couldn't detect it. Maybe if we can use those specific markers to pull down the vesicles, then probably we can do it.
I can send to you the nanobody against DPP6. We can discuss by email. And
yeah,
that sounds great. You know, another thing I've thought about is like that even if the pull downs aren't specific enough to really say we're like enhancing that much, like I think we could do like the exochip and look at co-localization with those, you know, at least would be a way to kind of understand with a small volume of biofluid potentially. That might be a way to do it. So, yeah.
Perfect. Thank you, Decio. Hi, Ed.
Hey, Emily. Really cool work. Very exciting. So the question I have is about pertaining to can these EVs serve as a stimulating source of antigen to the immune system? Particularly, you know, since they have HLA and PDL1 or maybe even on separate vesicles, but can these EVs interface with T cells directly, or do they have to be, you know, taken up and represented somehow by APCs? Or do you have any data on that, or do you look at that, or even just what are your thoughts?
Yeah, we haven't done we haven't done anything about antigen presentation, but I think I totally agree with you. I mean, I think, you know, you guys' data suggests that like antigens are there, right? Like we've looked a lot at proinsulin because I was really excited about proinsulin and EVs, and it doesn't seem like the actual proinsulin quantities like in global EVs change that much, like when you add like cytokines or inflammatory stress. But I think it also could be like there are differences like within these subpopulations, like maybe within the HLA positive ones, you know, and that, you know, there could be differences in how they get presented. For sure. Like I think that could definitely be, especially like when you think about like epitope spreading and stuff, it seems like that could be a mechanism there.
Yeah, it would be really interesting if you can find like neoantigens and things of that nature. Because, you know, the EVs can migrate to the draining lymph node and so forth, or be carried there by APCs.
Yeah, I totally agree. Thank you.
Perfect. Thank you, Ed. I'm looking for other questions, but I want to, Raghu, go ahead.
Yeah, I'm curious, Emily, why you had originally hypothesized that the double autoantibody positive individuals would have higher PD-L1 levels. Because I might have hypothesized it was single autoantibody positive.
Yeah, I guess, I mean, maybe it wasn't very carefully thought through. It's a fair point. I guess I was just thinking things are more abnormal, like it's going to be more revved up, right? In the context of multiple antibody. But I mean, I think like in retrospect, it definitely makes sense that, you know, if singles are earlier on in the process, or, you know, we know a lot of people get single autoantibodies and they stay there and they never move on, right? So it could be that aren't they doing that? It could be that those are the people that really upregulate PD-L1. And so they do okay. Yeah. So
I wonder, Emily, about the state of the individual when you collect these samples, because Ernesto, your exercise experiment was interesting, right? Because you're taking these firefighters at a resting state, and then they exercise, and if I've understood your data correctly, you're actually seeing a difference in this EV peak. So Emily, you're surprised, and you say, wait a minute, I don't see any difference, but do you need to account for something in these individuals? Do you need to challenge them in some way? Do you need to do something different in order to perhaps see some differences as you did in the exercise experiment that Ernesto did?
Yeah, that's a really interesting point. We usually collect our samples fasting just because we're so interested in all the metabolic markers. And, as you know, they all change, right, at different states of stimulation. So I think fasting is like a nice, a relatively easy to get time point. You're not having to do a bunch of these like stimulated tests. And then metabolically, it makes everybody the same, kind of. I think that's a good, like, is there a way that you could like stress people and see some differences? And I think very feasibly, if it's related to metabolic health, you could, that's a good point.
Yeah, if I can complement that. So one of the proteins that we see higher in the auto-antibody positive individuals is CD63. So we were just having an idea that maybe some of the vesicles are being secreted are different. And then one thing that caught my attention in Emily's presentation, that was kind of the control with the cells, but I can see there was like a band for CD63 was depleted after treatment cytokines. So I was just wondering if maybe the cells secrete more CD63. I was one of your slides, Emily. Sorry.
But Ernesto, you're saying these were autoantibody positive, no
Yes.
diabetes,
No diabetes yet.
Single or multiple?
I think it's mainly single. I
Okay.
don't have the information in my mind.
Yeah. Okay.
And then we haven't looked yet the peak height. Maybe there is a difference in peak height as well.
Interesting.
Yeah, that's an interesting point. In ours, it looks like in that blot I showed, the CD63 is lower in the cells with cytokines, but it's similar in the EV populations. But that's really what we were doing it for, was make sure it was enhanced in the EV populations. So I didn't think that much about it being changed in the cells. Go ahead, Raghu.
Emily, and Ernesto, CD63 we think of as being, associated with, exosomes and as a particular tetraspanin. But, there was a very interesting paper by, I think it was Jonathan Lowe in Nature Cell Biology that showed that CD63 is expressed on more functional and mature beta cells. From single cell sequencing analysis, and it's a very nice paper. And it got me scratching my head. What does that mean? Well, he told me when I spoke to him that CD81 seems to be the opposite. That as beta cells dedifferentiate or become less functional, there's more CD81. So to some extent, when you're looking at these tetraspanins on beta cell populations like you are, they might reflect, the state, like you see less CD63 when you throw on cytokines. That could reflect the functional state of the beta cells too.
Yeah, I think that's a good idea for looking at subpopulations too, right? Of the EVs, to think about it that way, maybe.
Terrific. Well, thank you, Emily. Thank you, Ernesto. Thank you, Raghu. We've had a wonderful conversation, lots of great questions and wonderful data. We hope to see an update and look forward to the paper officially being released, Emily and team. So again, thank you on behalf of everyone. We appreciate your time.
Nice to see everybody.
Thank you.
Okay.
Bye.
Bye, everyone.